you so much for inviting me to speak. It really is an honor and a pleasure to be here with all of you. What a great meeting. So I'm going to take you through some of the work we do at Sage, and I'm going to talk along the way a little bit about my own journey into this organization and into a non-traditional career in science, because I hear that is of interest to many or at least some of the people on the field or on the call. Okay. So we are at such an amazing time in biomedicine right now. There are emerging technologies coming online all the time that are allowing us to take deep, exquisite looks at an individual, at what health means and what dysregulations are causing disease. We can do this at the molecular level, at the cellular level, at the organ level, and increasingly we can do it for humans at the individual level as well as they go out about and live their life. And so we've moved really in the last 10 years to a world where we no longer need to bring an individual into a clinical laboratory in order to run a clinical experiment. We can now actually observe them in their own lived environment through collections of their medical records, through the interactions that they have with technology and technology platforms just on an everyday basis, and through the inclusion of sensors into their world, whether that's in their home or on their person or what have you. And so we really are able to get a much more holistic view of what it means to be healthy and how that can be altered by disease experience that we have. There's so much room for discovery in the world right now, but we continue to have a big issue in what we call the translational divide. So the scientific enterprise that we all have joined is really one focused on discovery. So if you think about the way this scientific enterprise is developed and how our careers progress, at every step of the way, whether it's getting a paper published or a grant reviewed or for your own personal professional development, the number one thing that we're all rated on is innovation. Did we make a new scientific discovery? Did we find something new that we could add to the scientific knowledge? And while this is really exciting and really important, it often leads scientists, it incentivizes scientists to think about innovation over reproducibility or robustness. And this means that when we want to think developing on top of some of these scientific discoveries, whether that's new methods for medical care in a hospital, or it's new pathways that we might want to be going after as drug targets or new biomarkers or what have you, it can often be very difficult to determine within the giant, wondrous world of all of these scientific discoveries, which of these are really reproducible and robust enough that we can build actions on top of them. It's not that those don't exist, there are lots of them, but it can be very difficult to find. And so over the last 10 years or so, open science has really come to the forefront as one approach that can be used to start to evaluate the reproducibility and actionability of scientific findings. So I expect many of you know, but for those of you who don't, open science is really a set of practices which are designed to support independent interrogation of our scientific works. In general, they're requiring scientists to work in transparent, reproducible, and accessible ways. And so data in the open domain, your methods in the open domain, your results in the open domain, so that others can see what you're doing and make their own objective, independent assessments of your work. And I think that this works on a couple of different levels. It is just inherent for any of us that if we need to show our work, we're going to double check it before we put it out there. And so we do tend to see that the quality of work changes when people are open to peer review, which effectively this is an extreme version of peer review. It also allows others to start to evaluate and build on one another's work. So I think open science, obviously, I think is really important. I'm at an organization focused on this, but it's not, it's necessary, but not sufficient for where we want to be going in terms of narrowing this translational gap. The other piece that we really need is collaboration. And so what I'm showing you here is a graph that came out of this great Nature paper about 2 years ago that just evaluated scientific outputs of research groups based on team size. And the title sort of says it all. What they found was that small teams are able to go fast, or they're able to identify new discoveries quickly, but it was really when larger teams came together with collaborations across multiple institutions and multiple disciplines that they were really able to objectively evaluate these various discoveries and start to develop actionable programs on top of them. So start to translate them from science into medicine. And so Sage Bionetworks, we've been around for about 12 years, and we were developed as a nonprofit really focused on the development and application of these sorts of open practices. And we're specifically working in biomedicine to do this. And we very intentionally operate as an independent organization that's not part of any one academic institute or pharmaceutical institute, because we often sit and act as a trusted partner who's aggregating groups of scientists who might in other conditions consider themselves competitors to work together in collaboration. I'll tell you more about the kinds of work we do. So far, this has been a little bit abstract, but first I just want to give you a sense of as I got here into this non-traditional career path. So as was kindly described in the introduction, right, here's my own career trajectory. I spent a fair amount of time training in academia. And then as I came out of academia, the first job I had actually was at Sage Bionetworks, where I joined as a scientist on their scientific team about 10 years ago. So I want to start by saying, well, why did I do this, right? I was definitely at a point in my career where I was thinking about academic research. My degree's in pharmaceutical chemistry, so clearly I was thinking about the pharmaceutical industry as a potential career path. How did I end up at a place that was neither of those things? Because at the time, there actually, it wasn't that long ago, but it was long enough ago that at the time there were a lot fewer options than there are now, which I think is really exciting for all of you. There's so many options of ways you can make your career go where you can really have an impact. I think that I had less choice at the time than you might have. So how did I end up at Sage? And it really comes down to the in my postdoctoral fellowship. And that work I exemplify here. So I was lucky enough to run a postdoctoral project that resulted in a first author Nature paper. And I show it to you here. It really doesn't matter what the topic is, actually. What I can tell you is I was using genetic and genomic analyses in cell systems to evaluate the impact of statins on cellular processes. And the goal really was to see if we could identify personalized medicine trajectories for people taking statins. What I learned in this project actually had a lot more to do with the process of science than it did with statins. So I had a finding, it was very interesting. And I looked around at the field around me, and there were a whole series of people who had data that I knew I could use to replicate my finding to evaluate, in fact, whether it was reproducible and something we might want to follow up on. And the hardest thing I had to do in this entire project was convince those other people that they wanted to partner with me and that they wanted to allow their data to be used to help ask the question that I was trying to ask. And it was a question of data accessibility, and then it was also a question of methodology. A lot of the methods we were using at the time were pretty new, which means they weren't yet standardized. And so there was a lot of back and forth that had to go on to evaluate the validity of the methods that we were using. And so what I can tell you was, it's very good for your career to get a first author Nature paper, but to this day, I don't actually know if this observation that I made is actually reproducible, sufficiently reproducible that one would want to take an action on it, because I simply could not convince enough other members of the field to buy into my hypothesis and join me. And the data was not accessible in any other way except by individual pairwise partnership. And that was pretty disappointing. And so as I came out of the postdoc, what I realized, I realized that these sorts of issues of data accessibility, methods validation, and broad scale collaboration to really evaluate, this is an innovative finding for which I couldn't assess the reproducibility or actionability of it. Those problems were far more widespread and important to solve than any individual biological hypothesis that I myself might actually want to go after. And at the time, Sage Bionetworks was just getting started, and they were looking to address exactly these problems. They're effectively wanting to innovate in the way that we do science rather than in any one specific scientific question itself. And I decided that that would be a place where I myself would find a lot of value and I could make a lot of impact on the field. And so I decided to take a chance on what was at that point a tiny nonprofit startup, effectively, to see if I could have an impact in the field. So I want to take a pause and say, if that story resonates to you in any sort of way, in the abstract, first, not even in the specific, but in the abstract, where you are seeing that there are interesting components of what you are doing that you might actually want to follow up on, rather that might be interesting to you to follow up on in your career, there are tons of places where you can start to do some really interesting work. And so I put here a series of sectors where you might want to consider career prospects or job opportunities. There are increasingly a whole series of independent research institutes that are blooming up all around the world that focus on research outside of the academic setting. And usually they're developed by someone who has a vision for a very specific type of research that they think can be done, and they find this to be the right approach. So as I said, for Sage, we very explicitly became our own nonprofit research institute because we knew we had to be a neutral party in order to be able to connect the individual collaborators that we wanted to connect. I've listed here a series of research institutes. All of these are in the city that I live in, which is Seattle, Washington. And I just wanted to sort of drive home the point that there are lots of institutes out there doing very interesting things, and you can find them probably just down the street from where you are, but certainly across Canada and the US and around the world. Another place that I have seen people do really great science is actually by joining an instrument or an assay company. When genotyping, chip genotyping, and then sequencing started to come alive 15 years ago, Illumina was an amazing place to work because there was access to new assays and huge data sets that nobody else had. Another example I've put here is a more recent company, Care Evolution, which does remote clinical trial assessments. And again, by being the provider of the instrument or the assay or the technology platform, you end up with access to a huge amount of data that allows you to really understand its scale, how such approaches can be used, where they work really well, where they don't, and you can start to see broader trends that you don't see in any one individual experiment. The third place I might direct you to consider is diagnostic companies, and these are exploding right now. The concept here, again, is that if you can collect data on humans across multiple medical centers, across multiple countries, on multiple diseases, and aggregate them into a single database, the power of that for machine learning will just explode our ability to understand human disease. And so a lot of these diagnostic companies, and I've listed 3 here, and I explicitly picked these 3 because amazing scientists that I know who were in academia have left academia to run the scientific teams on these 3 companies in the last 4 years. And so a huge loss for academia, but all of those people said, actually, I can't get the scale of data I need in academia to do the kinds of research I want to do, and I can within the context of these companies. So if you're a big data person, I really recommend thinking about these sorts of approaches. The 4th sector I wanted you to consider was funders. Some of the smartest, most impactful people, scientists I know in the world are the ones not running the experiments, but the ones at the foundations who decide where the money gets spent. And they are guiding the entire scientific enterprise in the direction that they think is right for the field. And it is an amazing power, and you can have a huge impact by thinking about joining in and being a funder. Journals are another place where you can really impact the field. And then the last 2 here, general tech companies and the government, I think are really important as well. So my company or organization, we hire data scientists, we hire software engineers, we hire biologists, and we hire data ethicists and regulatory scientists. And what we've increasingly found over the last 5 years is we'll be interviewing data scientists, and we'll say, you should come work for us. Sure, we're a nonprofit and we can't give you, stock options and huge salaries, but the reason you would want to come for us would be for the value proposition around impacting health and society, and that we are a place where you can really do that. And increasingly, I've had data scientists say to me, Apple provides me with the same value proposition, or Microsoft has started a, AI for Good group, and they have mission-driven sectors within their large tech company that are doing the same thing, and I can go there and do the same sort of work and feel just as good about it. And that's pretty amazing. I mean, there's huge power to being inside one of the tech companies if you're trying to impact or operate in the kind of field where the science you do is tech driven. So I wanted to put all of these out here for you to consider as you're thinking about career options. There's lots of ways you can go. Okay, so now I'm going to shift back to what it is we do at Sage to give you a little slice of what that might feel like, being at a nonprofit research institute, which has a directed mission that it focuses on. Okay, so we started at Sage with this concept that we needed to be doing open and collaborative science in order to support the translation of biomedical research into medicine. The place we started was that in order to effectively do this, it's very simple, cut and dry. All data needed to be put in the open domain, and that this would benefit society and science by allowing us to independently evaluate each other's results, but also to reuse that data in all sorts of ways that might not have been anticipated when it was originally generated. And there is a huge benefit to open data. One clear example just from the last year during the pandemic was the open databases that were being used to deposit the viral sequencing data coming out as COVID-19 swept around the world. And the ability to actually track the virus and the mutations of the virus as it spread across the world really was very helpful, both on a scientific side as well as profession, to understand what to anticipate where. But even this very simple case of an open database that's used for viral sequencing actually has some nuance to it around this concept of open. So anybody who might be following this saw a whole series of commentaries and papers start to come out about a month or two ago, indicating that not all researchers were in favor of the use of a completely open database. And the reason for this was scientists with greater resources, private resources, were able to capitalize on the use of the open databases more than researchers who had fewer resources. And so the particular example that came up was researchers in the Global South who said, they spent all of their extra time making their data accessible, and scientists in the wealthy nations were then using this data, performing analysts, having the observations, and actually getting the credit, and that it was undermining their efforts. And so they are in favor of a more restricted form of sharing that ensures that they get attribution for the portion of the work that they're doing. And I think that the question of attribution, and actually the question of how we as scientists can think about value for contributions is important as well, right? We continue to operate in a world where we value the analysis over the generation of the data, and in that inequitable case, one has to think very, very clearly about how you do attribution, particularly in cases of open data. The other reason we have started to consider methods other than purely open data sharing is the following. In human research, most of the data we need to answer impactful clinical questions is simply not open. And often these are for reasons of privacy or security or confidentiality. It's because the data might be coming out of somebody's medical record, or the data is proprietary in some means. And so what ends up happening is that if we focus entirely on open data, we often simply don't have enough data to be able to effectively answer the questions we want. And so over the last 5 or 8 years, our organization has shifted from this concept of completely closed data or completely open data to a much more nuanced landscape where data is openly available in an approach that is fit for purpose. So what is the science you're trying to do with the data? Who does it need to be open to in order to do that? How open does it need to be? And there are all sorts of systems that we and others have put in place, rely on a combination of technology and governance to enable open science on data that is not itself always completely open. And I'll just give you one example of what this looks like so that you know more, you have a better sense of what I'm talking about. Here's a whole series of governance structures one can put into place to allow these sorts of things to happen. And here's a very specific example of a method one can use to perform open analyses. This is a data enclave or a sandbox. And in this case, data donors a data steward sits at the center of this. This is often the role that my organization plays. They negotiate with data donors to understand the conditions for data transfer and use. And then the data steward holds the data as a host, as a steward for the data, and works with data users to ensure that they are in fact operating under the very specific terms with which the data can be used. And so for the data enclave approach, the data is put in some sort of a cloud-based sandbox, and anyone who wants to use the data actually has to come to the sandbox in the cloud and use it there. They may not download the data. In some cases, they can download a synthetic form of the data, but not actually the data itself. So a great example of this actually from last project we had going on in the US called the National COVID Cohort Collaborative. We do not have a national healthcare system. This causes all sorts of problems, which you have solved through policy rather than through data stewardship. So that, that's probably a better option. For us, we are stuck with a whole series of medical centers that don't actually share their data. And when we try to look at something like the impact of COVID on our patients, trying to build either diagnostics or treatment plans based on the evidence coming out in real time, we simply actually don't have a research resource that allows us to do that. And so we were able to create a data enclave, this N3C, which was run by the NIH, as a centralized, secure place to access COVID-19 data coming from patients across 80 medical centers, simply by putting in place the governance and the technology to support this. So that's the first part of this, right? I said that Sage is interested in science, open and collaborative science. The first piece, the open piece, is about creating technical and governance solutions that allow data to be available to those that need to work on the data. The second piece is about collaboration. I'm sure that you have experienced in your own careers this very simple statement that scientists already work in trusted communities. And so what we've continually found is that when we want to do scientific evaluation, when we want to evaluate the reproducibility or robustness of an innovative new finding, it often is an established community of scientists who are already working together within their own network, who will come together to do that sort of large-scale objective evaluation. And so we have started to embed into a whole series of research communities where we can help support them in doing objective evaluation on the open data that they have to come to these sorts of trusted findings. And so I want to take you through one example of what this means, again, because it means more in the specifics than it would in the abstract. And this is an example from Alzheimer's disease target discovery. So again, here in the US, it is a time of increased investment in the field of Alzheimer's disease. Congress, our government has allocated additional funds to our NIH, our federal health research funding organization, over the past 5 years, which has tripled the amount of money that we have accessible for Alzheimer's disease research. In addition, a whole series of private funders are investing themselves. They're investing their own private money into the field. This is a topic that lots of people are trying to solve. And that's really exciting. This happened last century for cancer. It's starting to happen now for Alzheimer's disease. But a big question that we have is actually, how do we ensure that those large are actually catalyzing new science, new innovative science, science of things that are actually understudied right now, rather than simply more money to study in more detail the things that we already know. And so I exemplify that with this graph. What I'm showing you here on the x-axis are all the genes that are known to be linked to Alzheimer's disease. And on the y-axis, we're looking at the number of publications that have come out on each of those individual genes. And so what you can see is that, in red, I'm showing you the publications that have come out in the last few years. And effectively, you can see that those continue to be on the same few genes that we were already studying 50 years ago. So this is another sort of externality of the way that the scientific enterprise works. Grants and papers are peer-reviewed, and often they're done so in a fairly conservative manner where projects which are working on something which is already known to be of high potential value will be upweighted over things that are working in unexplored areas. And so people tend to work on the same things rather than on new things. And this This is a problem when we're thinking about something like Alzheimer's disease, where we know, because it's a heterogeneous disease for which we don't yet have any medications that impact the biology of the disease, possibly with the exception of the approval of aducanumab last week, that we really are going to need to know more about understudied mechanisms and to be able to effectively treat this disease. So this is the opportunity. We use open collaborative approaches to incentivize scientists to move down into this area. And there's two things we need to do. One is we need to make sure that we have we understand what the full complement genes are that are linked to Alzheimer's disease. So effectively we need to expand the x-axis, and then we need to try to shift research away from those well-studied disease targets and into this long tail. It's not to say we shouldn't continue to study those important mechanisms, is that we need to complement them with others. And so we got involved in an open collaborative project that was working to do exactly this thing about 6 years ago. The project was pretty simply put together as follows. A whole host of different research groups were basically tasked with doing the same thing. They were tasked with generating multi-omic data on human samples, running complicated approaches to model the molecular state of disease within those human samples, use that to identify targets, and then evaluate those in some sort of preclinical model. And the beauty of this was we knew that this approach was likely to be fruitful, but there was not yet a standardized method for doing any of these things. And so by funding multiple people to take their own shot on goal, as it were, at this problem, and then aggregating the data, the evidence, and all the digital outputs that come from these groups together, we can take a look at where we see consistency, independent of method or data source, et cetera, around emerging targets that we might want to go after. And so my group was responsible for the coordination of the data and the scientific analysis. These individual groups was responsible for the innovation in the approaches that they were taking. And just to give you an example of what kind of data I'm talking about here, I wanted to show you this. There were 3 main data sets, which are in the headers here. Each of these are human brain samples, postmortem brain samples, on which we are starting to layer multiple levels of omic characterization, because we really wanted to get a sense of what the molecular state was in a healthy brain, in a diseased brain, and importantly, what the steps are in that progression from one to the other. And so my team has put together a technical infrastructure, a cloud-based infrastructure, of which this is a graph describing, that can be used to ingest the data, process the data, analyze the data, and evaluate results in aggregate across all of these groups. And the data, of course, is not available just for those in the consortium. In this case, we are able to make the data open. And so there is a portal where the data is openly accessible for anybody to use. So if you have any interest in the sorts of data I was just talking about, please come here. It is available for you. So as I said in the beginning, the first step in any sort of open evaluation is making the data open. We were able to do that. The second step is to start to evaluate the methods that are being used across each of the groups. And so the sorts of complex network-based analyses that each of these groups are doing take in a whole series of inputs into modeling itself. Although the network modeling is what I'd call sort of homegrown, meaning each group has their own method that they use to do network modeling, the inputs are actually pretty consistent. And so I show you some of them here. And so the first thing we did was, as a consortium, we came together and we did a wide-scale standardized analysis of each of these types of analyses to allow us to come up with a community validated or a community vetted output, so that we knew that the inputs to the network modeling approaches were things that were themselves robust and well validated. This was highly useful, but can only be done for the kinds of analyses for which there is sort of standard practice in how you might go about doing that. When it came to the network analyses themselves, because these are not standard practice, there is not one sort of universal truth for how one might want to do that. And so in that case, we do one giant meta-analysis. What we need to actually do is start to evaluate each of the methods that each of the groups was performing relative to each other. And so we ran a large-scale consortium-based analysis, which looked at the outputs, the target predictions coming out of each of these groups across every single one of the network algorithms they were using and across every single one of the data sets that we had access to. And the goal was to be able to say, what are the outputs? How do these results look independent of algorithm or data set? And what we found is as follows. So on the left, I'm showing you a heat map that represents the gene or protein-based modules that we found to be dysregulated in human brain in Alzheimer's disease. And this particular data set, or this particular graph, is coming from transcriptomic data, but we have replicated this in proteomic data. And what we basically see is that there are 5 big blocks here, and those represent 5 different biologies that were highly impacted by Alzheimer's disease, independent of what algorithm was used, what data set was used. We consistently see these same 5 domains of biology coming up as important. And when we mapped onto these 5 domains of biology, those gene sets or biological pathways that have already been reported to be important in Alzheimer's disease, we see that literature maps to 3 of the 5 domains, but actually 2 of the domains are really understudied in the literature. So these represent some interesting new places to start to look. And so each of the groups within this consortium had gone on to prioritize biology that they were going to study themselves. And you can see that they all predominantly focused on biology in these 2 newly discovered understudied domains. So this is great. This is, to that graph I showed earlier, we're expanding the x-axis, we're identifying new areas of, and these individuals within the consortium are headed that way to study those things. And I can tell you one of these biological domains is related to myelination, and one of these biological domains is related to intracellular processes that are actually pretty general to a whole series of brain or any cell type related to protein and RNA regulation. Realizing that neither of these domains had come up consistently in the literature before, and in particular, they weren't really coming up in any of the large-scale genetic association studies or sequencing studies that have been performed now across the world. And so we were able to then start to take a closer look at what we might know about the biology of these 2 areas, given these meta-analyses that we were able to do. And one very interesting thing we found was that there was, in fact, some significant heterogeneity in the manifestation of these 2 biological processes in Alzheimer's disease. And in particular, we saw a big difference between male samples and female samples. And so this sort of heterogeneity is really well known. It's well documented clinically in Alzheimer's disease. There are big changes, big differences in how Alzheimer's disease manifests in men versus women. And you can imagine if you're doing a large-scale genetic association study, often you're usually correcting for sex versus specifically studying, seeking out sex-specific genetic associations. And so if you're correcting for sex, you're actually not going to see anything. You're only going to see the things that are consistent across everybody. And for this disease, and actually many brain diseases where we know that we categorize disease based on a common symptomology, but actually probably what we're studying on the molecular level is a whole series of different diseases that just manifest the same. It's often the case that when we do large scale sort of aggregate analysis, we miss a lot of the nuance. We miss a lot of the biology that's actually only seen in some of the samples or some of the individuals and not everybody. And it's only by pulling all of this together that we're able to actually start to see some of those nuances. So this is all well and good, but so far everything I've told is computational. So we can find methods of open science, of collaboration on computational analyses. What we really need to do next is actually catalyze experimental evaluation of those hypotheses. So how do you convince an experimentalist that they want to spend the next 3 to 5 years of their life studying a hypothesis that came out of a computational analysis? You have to be able to demonstrate pretty robustly what the evidence is in support of that hypothesis for anybody really want to make that bet. And so we're wanting to catalyze that. So how do we go about doing that? Well, the very first thing we're able to do, again, is to use open science approaches to make sure that all of the evidence around every single one of these target predictions is available, accessible, and understandable to biologists or clinicians who might want to evaluate it itself. So there's a step of translation from the world of a bioinformatician to world of a biologist or an experimentalist. And so we are providing a service. This is a web-based service called Agora that provides that sort of documentation around the evidence for these targets. The second thing we're doing is to start to catalog each of these understudied target hypotheses. So aggregating the signals or the computational signals or the targets that were found into individual or broader hypotheses that we may start to think about biological mechanism around. And then the third thing, actually there's 4 things. A third thing that we're doing is to start to use the computational models of human disease that we have and doing comparative evaluation with other species, and in particular those species that are used in experimental models. So if you want to study the effect of Alzheimer's disease on myelination, we can know exactly what that pathway looks like there. Then we can look for that pathway and an impact of Alzheimer's disease on that pathway. In this case, we're looking across many different mouse models to ensure that a researcher who's wanting to do a mouse study is picking a mouse model that actually recapitulates on a molecular level the disease as it's seen in the human. So this is a really nice way to be able to make sure that our experimental models are well matched with the human condition or the specific molecular pathway in humans that we're wanting to look at. And the last thing we're doing is a partnership with the Structural Genomics Consortium. So this is a Canadian-led group led by Alan Edwards, who's at the University of Toronto. The Structural Genomics Consortium over the last 10 years has been developing structural biology and medicinal chemistry outputs. So crystal structures, purified protein, validated antibodies, molecular probes, and putting them in the open domain for anybody to use. And the hypothesis here is pretty simple. It's simply that a lot of times researchers might be interested in an understudied protein, studied biological pathway, but they lack well-validated, easy-to-access reagents to actually perform that research. And so just the activation energy of starting to study something understudied is really sizable. You have to actually build your own reagents, validate them before you can start to do the biological experiment that you really want to do. And our hypothesis is that if we simply ourselves make and validate reagents and put them in the open domain, it will in fact catalyze research into understudied areas. And the Structural Genomics Consortium has a long history that demonstrates that in fact that has been the case in many other areas that they've worked. And so we've partnered together in Treat AD, which is going to spend the next 5 years building high quality validated research tools and putting them in the open domain, and then publicizing these so researchers across the community can pick them up to use them to study these biological processes within Alzheimer's disease, but frankly, you know, in any other condition that they may be interested in as well. So we often get questions about this. Are we putting the cart before the horse? We're taking, you know, what we're calling targets, but really these are computational hypotheses that have not been experimentally validated, and we're making a whole series of drug discovery reagents, including small molecule inhibitors. But in fact, our hypothesis is that in order to really effectively evaluate any biological mechanism, one needs to have access to high quality tools. And so the target validation and the drug discovery actually need to happen in an iterative process together, and not one and completely finishing one before you do the second. I wanted to show you what targets we've picked. I put these up there. This is the first round of targets that we're building validated reagents against. I put these up here in case any of you happen to be interested in any of the targets that are on this, any of the proteins that are listed here. Please get in contact. We are building experimental reagents. We would love to provide them to you so that you can use them in your own experiments independently and in whatever way you may want to. Okay, so in summary, I hope that what I've been able to exemplify for you here is that we are able to use open collaborative scientific approaches to start to bridge the translational gap that takes us from the exciting academic world of discovery into the world of development, whether that's drug development or development of clinical care practices. And we open science approach helps to speed this process. It helps to provide community level verification of research outcomes that lowers the barrier for those individuals who work on the development side to start to pick up discoveries and move them forward. And that is when I started this job 10 years ago, exactly what I was hoping to do. And it's been pretty rewarding to see in one example after another how these approaches can actually help to do that. I'd like to just end by thanking the team at Sage Bionetworks. We have a pretty amazing group of people, and I feel really lucky to work with all of them.